Hello guys, this is a new series. In this series, we will be working with whole biology textbook. In this series, we will only be able to finish 5 chapters. Chapter 1, Types of Transporting. Chapter 2, Photosynthesis. Chapter 3, Cellular Respiration. Chapter 4, Taxonomy. Chapter 5, Viruses. Let's hurry and start chapter 1. Chapter 1, section 1, passive transport. In the passive transport is the movement of substances across the cell membrane without using energy. So what this means is when substances, molecules, atoms actually pass through the cell membrane without using any energy by the cell. So the cell does not expend any energy for transporting molecules by passive transport, okay? So simply, this is cell membrane. Those are atoms and they are passing through in the cell. This is out of the cell and this is in the cell. So they are passing through in the cell without any input of the energy by the cell. So the cell does not expend any energy, okay? So those are two types of passive transport, but passive transport have 4 types, okay? So first of all, diffusion, second osmosis, third facilitated diffusion, and fourth one is diffusion through ion channels, okay? So first of all, we will be starting about diffusion. What is diffusion? Diffusion is the simplest type of passive transport. So the simplest type of passive transport is diffusion actually. It's the movement of molecules from a higher concentration to an area of lower concentration. What this means? So I will teach you this in this figure. What do you think? Which side is the higher concentration? So this side is the higher concentration. Why? Because there are more molecules here than here. So for example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 molecules are here and 2 molecules are here. Actually, they must be the same, okay? They must be the same. So diffusion is gonna take those molecules to the process to make them the same. So after diffusion happens, actually those molecules, 8 molecules or 7 molecules are going to be here and 7 molecules are going to be here after diffusion, okay? After diffusion. So, okay, so simply diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. We can say also passive transport is the movement of molecules from an area of higher concentration to an area of lower concentration. Okay, this differences in concentration of molecules across a space is called concentration gradient. Okay, this is the differences. For example, here there are 18 or 16 molecules and here there are only 2. This differences is called concentration gradient. Okay, concentration gradient. Okay, so diffusion across cell membranes. As you see, before diffusion, the particles outside was more than the particles inside. As you see, for example, 50 particles outside, but only 10 or maybe 20 particles inside the cell, okay? But after diffusion starts and finishes, the molecules in the cell and the molecules out of the cell are same, okay? Are same. So here, the concentration gradient, there is no concentration gradient actually in this one, okay, because they are same, okay. So what if they're both same? What we call that? We call that equilibrium. What is equilibrium? In absence of other influences, diffusion would eventually cause the concentration of molecules to be the same throughout the space. So what this means, diffusion after diffusion, equilibrium comes. Okay, why? Because diffusion will just gonna take the molecules through the process of making both sides same. Okay, so if both sides are same, we call that equilibrium. Okay, we call that equilibrium. Okay, in the absence of other influences, diffusion will eventually cause the concentration of molecules to be same throughout the space the molecules occupy. Okay, when the concentration of molecules of a substance is the same throughout the space, a state of equilibrium exists. Okay, at equilibrium, the random movement of molecules continues, but because there is no concentration gradient, equilibrium is maintained. Okay, so what this means? So in every transportation, molecules are randomly moving. So, for example, this molecule is coming here, this molecule is coming here, and this molecule, they're randomly moving. Even after equilibrium, this movement continues. But because both sides are the same, there is a really less chance for one molecule to get this side. And if one molecule gets this side, so the another molecules come back to the other side. So every time the equilibrium is going to be maintained. Okay, so here the second type of passive transport, which is osmosis. Osmosis is the movement of water molecules. But before of this, I'm just gonna tell you what is solute and a solvent. Okay, a solution is composed of a solute and solvent. What is solute and what is a solvent? Solute is sugar, and solvent is water. Okay, osmosis is the movement of water molecules from an area of higher concentration to an area of lower concentration. Okay, so we said that passive transport is an movement of molecules from an area of higher concentration to an area of lower concentration. Therefore, osmosis is a type of passive transport. Okay? Direction of osmosis. So we said osmosis is the movement of water molecules from an area of higher concentration to an area of lower concentration. Okay, is the movement of water molecules. So what if the water molecules move in or out? We don't know which side is the higher. Okay, I'm just gonna tell you now. Maybe you don't understand right now. I will show you the figure. Okay, direction of osmosis depends on the relative concentration of solutes on two sides of the membrane. So we said, what is the solute? The solute is sugar. So when solute is in the water, so the water particles may move. Okay, I'm just going to teach you now in the figure. So depends on the relative concentration of solutes on two sides of the membrane. When the concentration of solute molecules outside the cell is lower than the concentration of solid molecules in the cytosol, solution outside is said to be hypotonic to the environment or to the cytosol. In this situation, water diffuses into the cell until to be equal. Okay, I'm just going to teach you this in the figure. Okay, so what is now happening really here? Osmosis is the movement of water molecules, not the movement of solute or sugar molecules. So those are sugar molecules, okay? Sugar molecules and water molecules. These little blue circles are water molecules and these are sugar molecules, okay? So before osmosis, a figure is like that, okay? The figure is like that. But after osmosis, something happens like this. What this means? Just think logically. This side is higher concentration or this side. Think logically, okay, guys? Don't think about the solutes. Think about the water molecules. So of course, this side is the higher concentration, right? So some molecules go that side, but what is happening actually? So here, just think logically, which of the sides have or has the more chance to get to the another side? What do I mean? For example, this side. This molecules move randomly. It heats this one, then heats this one, then heats this one, then heats this one, and 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 then heat this one, and then maybe go to the other side. But those molecules goes like this and heats this one and comes here. For example, this molecules, if comes like this and goes here, and if this molecule comes here and it goes here, this molecule come here and goes here, so it goes to the other side. So the chance of those molecules to get the another side is more better, okay, more. So how to know if this is a hypertonic or a hypertonic or a isotonic, okay? So when the concentration of solid molecules outside the cell is lower, okay? So what this means, for example, let's say this is outside of the cell and this is inside of the cell. This side has more solutes, has more sugars, okay, has more sugars. So if this side have more sugar, so this means that this is hypertonic, not hypotonic, not hypertonic, hypertonic, because this side is more. So let's say this is the outside of the cell and this is the inside of the cell, for an example, okay? So what happens, the solutes are really less amount in comparing to the other side of the membrane. So what do we call this is, we call it hypotonic to the cytosol. So this side is hypotonic to the cytosol actually, okay. So when the concentration of solid molecules outside the cell is higher than the concentration of solute in the cell, the solution outside is said to be hypertonic to the cytosol. So as I said, let's say this is out of the cell and this is inner of the cell. So if solid molecules are more out of the cell, so we call this hypertonic. But if the solid molecules are less, for example, this is outside of the cell, let's say outside the cell the molecules are less, okay, so what we call this is hypotonic. So when the concentration of solid molecules outside the cell is higher than the concentration of solid molecules in the cytosol, so actually this is now hypotonic, okay, if we call this side the inner cell, okay, hypotonic. So guys, logically think, what happens when we are in a hypotonic environment? In a hypotonic environment, water moves into the cell more. Why? Let's just understand. Because in a hypotonic environment, the solid molecules outside is lower. So here, the solid molecules outside, this is outside of the cell, okay? This is outside of the cell. An example. This is outside of the cell, so the movement of water molecules is easier and more, they have more chance to move to the other side. So if these molecules move like this, so they both go to the other side, okay? They both go to the other side, okay? So what this actually does is water moves a lot into the cell. Water moves a lot into the cell. Oppositely, for hypertonic, let's think that this is outside of the cell and this is inside of the cell. So inside of the cell, water moves out, okay? Because again, the chance is more for water to move out, okay? So diffuses out of the cell unital to be equal, as you see in the hypertonic, and in the hypotonic moves into the cell. So how to solve this problem? When water moves a lot into the cell may just burst, and after bursting, you know what happens, it will just boom up, okay? So how to solve this problem? Actually, before telling you how to solve it, I'm just gonna show you isotonic. What is isotonic? So when the concentration of solid molecules outside and inside the cell are equal, the situation outside is isotonic to the cytosol. In this situation, water diffuses into the cell. So simply isotonic are the same. Solid molecules in the cell and out of the cell are same. It's simple. Okay, I'm gonna pass this one here. Now, how cells deal with osmosis? As I told you, how to solve this problem. How to solve a problem when water diffuses into the cell a lot. How to solve it? How to, don't let the water just burst a lot the cell. So what we are gonna just do is simply using some enzymes in the cell. So what we're doing. Let's just see. Some organisms live in a hypotonic environment, for example, paramecium. Live in fresh water, which is hypotonic to their cytosol. So what does hypotonic means? Hypotonic means water moves into the cell a lot. Water moves into the cell a lot, okay? So when water moves a lot into the cell bursts and get bigger, and eventually, if nothing controls that cell, it will just boom, okay? So if we think that all of our cells are in water, so how can we solve this problem? We're not just gonna let our cells to die, all of them, okay? So what our cells are gonna do, they are gonna use contractile vacuolysis. Okay, water constantly diffuses into the organisms. They have contractile vacuolysis, which are cell organelles that remove excess water. When water diffuses into the cell a lot, contractile vacuolysis starts to work. So this contractile vacuolysis pumps out the water, okay? Pushes out the water. Go out. I don't need you, okay? You're just gonna make me die, okay? Just go out, okay? Contractile vacuolysis is gonna tell the water, okay? Which are cell organelles that remove excess water. Most of the time, plant cells also live in a hypotonic environment, okay? What about the plant cells? In the animal cells, contractile vacuolysis starts to work and does not let the cell to die, okay? What about plants? Plant cells also live sometime in hypotonic environment. Water moves by osmosis into the cells, but after the water moves into the cell will think, that's it, I don't need more water, okay? Let's just close the doors. The cell wall is just going to be strong enough to take the pressure that water molecule exert against the cell. So what is here now actually? The pressure that water molecule exert against the cell wall is called turgor pressure. So what is here actually? The cell wall is protecting the cell, actually not letting the water get in a lot, okay. So the cell wall is strong enough to resist the turgor pressure, okay. The cell wall is strong enough to resist the turgor pressure. Animal cells swell and eventually burst in a hypotonic environment, okay. So when water diffuses a lot into animal cells, of course they get big and big and big, but the contractile vacuolysis does not let the cell die. It pumps out the water, okay. The bursting of the cell is called cytolysis. In a hypertonic environment, water leaves the cells through osmosis. So now here what we understand, if we are living in a hypotonic environment, so water diffuses into the cell and the cell gets bigger and burst, okay, burst. But what if you are living in a for example, hypertonic environment? Water leaves the cell and the cell gets shrink, okay. The cell gets shrink, okay. So what about isotonic? It's a normal cell, gets out and gets in normally, okay. Subscribe and don't forget to leave a comment if you have any question. Don't forget to check the description, and if you didn't understand anything, just comment me, and don't forget, check the description, it's really important. I will see you guys in the next video and starting section 2. See you guys in the next